Today in this class I am explain about rice genome project. Scientific name Oryza sativa, family Poaceae, number of chromosomes 12 pairs, 24 in number, and genome size 430 megabase, and variety of rice used for the project Nippon bear. And what is genome? A genome is an organism's complete sets of DNA, including all of its genes. A genome is an organism's complete sets of DNA, including all of its genes. The total amount of DNA of a single cell, that is haploid cell in case of diploid organisms, and the genome sequence of an organism include the collective DNA sequences of each chromosome in the organism. Next, what are genome project? Genome projects are scientific endeavor that ultimately aim to determine the complete genome sequence of an organism and to annotate protein coding genes and other important genome encoded features. And genome sequencing is figuring out the order of DNA nucleotides or bases in a genome, the order of A, C, G and T, that is adenine, cytosine, guanine and thymine, that make up an organism's DNA. As the first step in a systematic and complete functional characterization of the rice genome, the International Rice Genome Sequencing Project, that is IRGSP, Rice Genome Sequencing Project, has generated and analyzed a highly accurate finished sequence of the rice genome that is anchored to the genetic map. The IRGSP, formally established in 1998, and the sources of sequencing groups in 10 nations to obtain a complete finished quality sequence of the rice genome, and provided evidence for a genome size of 389 megabase and a total of 37,544 non-transposable element rated protein coding sequences were detected. A total of 2,859 genes seems to be unique to rice and the other cereals, some of which might differentiate monocot and dicot lineage. And the transposon content. Transposons means mobile genetic element or jumping genes. That is a DNA sequence that is able to move or insert itself at a new location in the genome. The transposon content of gene is at least 35% and is populated by representative from all known transposons superfamilies, and the availability of a complete and high quality map based sequence has provided the opportunity to study genome organization and evolution. Most importantly, the order and identity of 37,544 genes of rice have been unrevealed. The sequence provides the required ingredient for functional genomics and molecular breeding programs aimed at unrevealing intricate cellular processes and improving rice productivity. Next is why sequencing of rice genome. It is one of the most important crops in the world and feeds over half of the global population. And the rice genome is the smallest of the major cereal crop genomes at an estimated 400 to 430 megabase, and it has relatively short generation time and has relatively genetic simplicity and provide valuable information which will apply to the cereal crops and to monocots in general. And complete sequencing of the rice genome will provide valuable information on the effect of repetitive elements on genome organization and evolution in plants. And grass genomes, including those of rice, wheat, maize, barley, rye, and sorghum, share a large degree of synteny, making rice an excellent mode cereal. And rice is also the easiest of the cereal plant to transform genetically. First organism whose genome was sequenced in 1996, the first eukaryotic genome, that is Saccharomyces cerevisiae, was sequenced. The rice genome is 3.5 times the size of the Arabidopsis genome and the third largest public genome project undertaken to date behind the human and mouse genomes. Next one is feature of rice genome. Oryza sativa has between 32,000 to 56,000 functional genes, and each cell has 12 chromosomes, and on rice smallest chromosomes number 10 are about 3500 genes and more than 22 million base pairs, the links in the chain of DNA. And repetitive DNA is estimated to constitute at least 50% of the rice genome and as much as 70% of the maize genome. And next one is vectors used in sequencing. Vectors used for sequencing: bacterial artificial chromosome or P1 derived artificial chromosomes, that is PACS. It refers to plant artificial chromosome or phage P1 vector. Sequencing of the rice genome is being performed mainly from genomic back or pack libraries created from the Nippon bear variety, which was chosen as the common template throughout the IRGSP, that is International Rice Genome Sequencing Project. The International Rice Genome Sequencing Project began in September 1997 at the workshop held in conjunction with the International Symposium on Plant Molecular Biology in Singapore. Scientists from many nations attended the workshop and agreed to an international collaboration to sequence the rice genome, and it is finished in December 2002. The International Rice Genome Sequencing Project has evolved to include 11 nations, and the IRGSP working group composed of a representative from each participating nation. And the recent interim IRGSP meeting at Clemson University on September 19 and 20 in 2000 in South Carolina was the largest rice genome meeting to date and was attended by more than 70 scientists and administrators from Japan, Taiwan, Thailand, Korea, China, India, Brazil, France, Canada, and the United States. The meeting was organized by Roadwing USA and International Rice Genome Sequencing Project representative Clemson University and chaired by Ben Burr, and International Rice Genome Sequencing Project coordinator Brookhaven National Laboratory in New York, and Takji Sasaki, Program Director of the Rice Genome Research Program in Japan. And India's rules. India's rules. India, previously an unfounded member of International Rice Genome Sequencing Project, has a new rice genome program represented by Akhilesh Tyagi of the University of Delhi and Nagendra Singh of the Indian Agricultural Research Institute, and worked on chromosome 11. And last one is application. First one, understanding plant evolution and the differences between monocots and dicots, and improve efficiency of rice breeding and improve nutritional value of rice, enhance crop yield by improving seed quality and resistance to pest and disease and plant hardiness, and development of gene specific marker assisted breeding of new and improved rice varieties, and how a plant respond to the environment and with genes control various functions of plants. That's about the rice genome project. Thank you.